Hello all, I am Manoja Aravindakshin. I am a PhD research scholar at the Department of Computer Science and Engineering, Indian Institute of Technology, Kharagpur. Currently, I am a visiting doctoral fellow at Purdue University, Department of Computer Science. So today I'm going to give a talk on mathematical modeling of drug distribution in liver-on-a-chip device. So I gave this talk during a workshop for data-driven mechanistic models of complex biomedical systems at University of Birmingham in December 2023. So this is a collaborative work done with ESQ Labs, GmbH. So here is the outline of the talk. I'll be giving a brief introduction on liver-on-chip. Then we'll focus on the modeling mechanism, which is physiologically based pharmacokinetic modeling. Then we'll analyze the sensitivity of the model to various parameters and estimate the relevant parameters in the model. Finally, we will move on to the discussion and next steps. So what is liver-on-chip? So organ-on-chip is a kind of microfluidic cell culture equipment which reproduces the physiological and pathological characteristics of organs or tissues by reconstructing the structure and function in vitro. So liver is the main site of drug metabolism. So we are interested in liver-on-a-chip device. So in the figure, we can see a liver culture chip LC12 with primary hepatocytes in the scaffold. The cross section of the chip is shown with micropump and inlet and outlet of fluid. So we are not experimentalists, but we work on the data, the time series data that is generated using the chip. Now, what is PBPK model? So PBPK model studies drug pharmacokinetics by viewing human organs as separate compartments and integrating them based on physiological knowledge. The behavior of drugs, absorption, distribution, metabolism, and elimination can be simulated. So even toxicity can be simulated these days. Now, what is the challenge? So accurate prediction of in vivo characteristics of drugs is important in the development of new pharmaceutical compounds. So yeah, this is a challenge that is currently ongoing. Now we will move on to the model. So first we describe a single compartment model. So in this model, that is figure A, so in this model we have the whole liver which is considered as a single unit and clearance is the only parameter. This is the state of the art or the conventional approach. So what they do is they do the regression analysis on the kinetic data to find the clearance of the drug. So you can see VdC dt is equal to minus CLC in dot C. So CL int is a clearance, intrinsic clearance parameter. So it is modeled using a single differential equation, which is regression. Now, intrinsic clearance means the intrinsic ability of the liver to remove the drug in the absence of restrictions imposed on the drug delivery to the liver cell by blood flow or protein binding. So we have constructed a three-compartment liver chip model consisting of the plasma, interstitium, and intracellular compartment. So the plasma compartment, it mimics the dynamic flow patterns reflecting physiological blood circulation, which influence the distribution of drugs to the hepatic cells. And interstitium compartment represents extracellular space surrounding the hepatocytes within the liver on chip. And intracellular compartment that represents space of hepatocytes where drug metabolism and various cellular processes occur. So these three compartments are modeled using three ODEs. Okay, I'll explain the ODEs in the next slide. So these are the ODEs for the three compartments. So CP, CI, and CC are concentration of the drugs in the three compartments. And one important thing is we could only observe the drug in the plasma compartment. So dCp by dt. So Cp is the only variable which can be observed. So this is the system of linear ODEs and it can be represented in matrix form with solution C of t equal to x e raised to lambda t x inverse C0. So this is represented in the matrix form. and we can find analytical solution using eigenvalues and eigenvectors. Uh, Now let us see the parameters in the model. So these are the parameters of the three compartment model. We have K1, K2, K3, K4. They are comprised of a group of other parameters. So I'll go through few of them. So K water int and K water cell. So that are the rate constant for water exchange within the interstitium and intracellular compartment. Similarly, SA is the surface area and FU is the fraction unbound in plasma. P endothelial is the permeability between the endothelial layer. Now, moving on to sensitivity analysis. So, we are doing both local and global sensitivity. So, local sensitivity is we are analyzing the local sensitivity with respect to the intracellular concentration, since clearance is considered through the intracellular compartment. So, the local sensitivity with respect to CC by varying the model parameter pi is represented as this. And we normalize this by pi to eliminate the effect of units. We also do global sensitivity analysis, which is parameter independent, and we calculate first order sensitivity indices as a mean effect to measure fractional contribution of a single parameter, and then we take pairwise contribution, and finally we will have the total order indices which consider the mean and the higher order effects. Now for that we use Sobol sensitivity analysis from SALib, which is a package in Python. And we set a range of lower and upper bound for the baseline parameters. Moving on, so these are the results of the sensitivity. So figure A is local sensitivity and figure B is global sensitivity. So in the figure A, the blue bars indicate the intracellular concentration and the parameters change in the same direction. And the red bars indicate if the intracellular concentration increase, the parameter value decrease. Similarly, figure B is blue and orange bar represent the first order and the total order indices respectively. So what it says is the clearance parameter is the most sensitive to the output intracellular concentration. Some of the other important parameters are the endothelial permeability of the endothelial layer, surface area, and fraction unbound. Now moving on to the parameter estimation. So this is a snapshot of data of diclofenac. So we leverage the available data on liver metabolism or liver toxicity, mainly from Dosi and Samandaras et al. So we have the time points, varying time points, and then we have the concentration in the plasma for each drug. Now, PKSIM from the Open Systems Pharmacology suite, we derive the basis of the liver-on-chip and the critical parameters such as permeability and water exchange rate of compounds. Now, how do we formulate the optimization problem? How can we estimate the parameters? So, quadratic loss function is the objective function here. So, we minimize over P, yi observed minus yi model whole square. So, yi observed is the observed data, the plasma time series data that we have. And yi model is computed by solving the ODEs with initial condition and some bound constraints on the parameters. So this is the optimization problem, where n is the number of data points. Now, we use Nelder-made derivative-free optimization to find the minimum of the objective function, and we estimated the parameters clearance and surface area of the chip. So the initial value of clearance. So we run the algorithm with 100 random start values of the clearance in the range 0.1 to 10-fold of the clearance that is obtained from the regression of the kinetic data. And after we obtain the clearance value, we do an in vitro to in vivo extrapolation. That is, we scale this value to relevant human values, that is using the hepatocellularity of the liver and also the liver weight. And then these are compared to the human observed in vivo clearance. So some of the results, so 4 of the drug simulations are shown here. So the dashed line indicates the single ODE model. The green bold line indicates the 3 compartment model, and the orange dots are the observed data. And the error of both the models are being shown in here. So we can see that the error in 3 compartment model is reduced and it is fitting more close to the observed data points. And also we are getting a better clearance value compared to the existing conventional model. So is the overall results of simulation of the 14 compounds. So we have the compound names here. We have the estimated in vitro clearance from the model. These are the third column is the observed in vivo clearance, human observed value. Then we have the predicted in vivo clearance. So this is a state of the art conventional approach values, and this is the 3 ODE model values. So after scaling it to human values, so we could compare it with the observed in vivo clearance. And these are the fold error. So you can clearly see that the state of the art clearly under predicts the clearance. We predict 261, actually it is 561. So for lorazepam it is 14, we predict 7.89, better than the state of the art. Similarly for other values. Now we'll see correlation plot so that graphically you can understand how better is the 3 compartment model. So the line in between the black line, diagonal line, is the line of unity. So we have the predicted clearance in the x-axis and the observed clearance in the y-axis. And the drug names are represented using a dot. The colors are like ECCS classification, which says low permeability and high permeability drug. So, sorry. So this is the single compartment model. This is the 3 compartment model. Most of them are very close to the line of unity in here. It is merely a representation of the earlier table that we had. Now moving on to the discussion. So why is this research important? So we need accurate prediction of the in vivo clearance from the in vitro data, because if the understanding is not correct, then this can lead to, unwanted clinical trials, and it may lead to underdosing and toxicity, etc. Now, earlier work. So as we saw, the earlier work was based on regression, and they just had one parameter and one ODE to analyze the kinetic data. So what we did is we did a PBPK modeling using 3 compartment ODEs to estimate intrinsic liver clearance. And we find that the intrinsic clearance that we predicted is far close to human observed values. Now, this 3 compartment model has a better representation with 3 compartments, and that replicate the functionalities of the human liver. And also it fits most of the drug kinetic data. The discrepancy between observed kinetics and the model prediction for some drugs suggests that it may need to be further refined to describe the data effectively. Now, what may be the key factor that contribute to this is the insufficient drug concentration within the intracellular compartment. So one solution is that we could increase the parameter surface area of the liver chip, uh, and that could result in a better fit of kinetic data. So this also shows that physiologically, what it shows is that increasing intracellular concentration allows more compounds to flow into the intracellular compartment. So if we have a way to measure intracellular concentration, we could further improve the model. So the next step, so as I said, measuring intracellular concentration is very crucial for validating and understanding whether the model needs to be refined or the biological model parameters may need to be more carefully determined. So we might need further experiments to improve the accuracy and reliability on the liver chip model. So as we refine the parameter estimation of this 3 compartment ODE, we could develop a digital twin of liver chip that can benefit pharmaceutical research and development. Thank you all. So I acknowledge Dr. Christian Mars, Professor Alex Pothen, and Professor Chittaranjan Mandal. And here are a few extra slides for calculation. So in vitro to in vivo extrapolation using liver weight and hepatocellularity. Also, I have a slide on Sobol sensitivity analysis, which is a global sensitivity analysis that we have used. Thank you all.